first time I've been able to go to the defense of one of my students in person. So I felt like I should say something. And obviously, I've been so lucky to have Ron in the group and basically have him come incredibly wonderfully vetted and trained from 2 other members of the committee. So it really was like one of the luckiest things that happened to me, that I was able to find such a good student so quickly on arriving here. And you know, you've been here much longer than I have and knew a lot about Stanford. You know, aside from science, like just knew like the ropes of Stanford better than I did in a way. So like sort of helped me learn how to fit in partially as well. So that, you know, I just have learned a lot over the years, and we've done a lot of really exciting and interesting science, and have sharpened a lot of notions and continue to do so. And I'm hoping we'll continue to do that in the future. I know most of you here have heard some of the things that Iran has worked on, but I think you'll be really just amazed to see the whole panoply of it together, and how much of a kind of take on doing neuroscience and computational neuroscience it actually is, in a way that's really unified and powerful. So I'm sure that there's many things I could say, but I don't want to take too much time later. So
yeah, thank you so much. I appreciate it. Yeah, so thanks everyone for coming. So today I'll be providing it in my defense, most of my work related to applying what's called goal-driven neural network models to systems neuroscience questions. And yeah, and in the interest of time, I'll take questions at the end of the presentation, but feel free to drop them in the chat as I go along. Yeah, so humans and animals exhibit a range of interesting behaviors in dynamic environments. And unlike me, they're able to juggle, do gymnastics, mountain bike in uneven terrain, and chase fast-moving prey. And as neuroscientists, we're interested in how the computations in our brains actively reformat this dense sensory information to enable these behaviors. And in order to be able to carry out some of the previous examples, we would likely require some level of scene understanding involving object-level classification, segmentation, and localization, to name a few, as well as some 3D understanding involving, say, estimating depth and normals. So how might we go about studying the means by which our brains might actually implement some of these computations? Initially, the only possible way towards these types of questions was a bottom-up one, where we build up the necessary elements circuit by circuit and neuron by neuron. But only in the past decade do we actually have models that can start to perform some of these visual behaviors with what are known as convolutional neural networks being able to perform image classification on data sets consisting of millions of images, with the ability to then transfer downstream to a variety of visual tasks. And these models are actually inspired by decades of observations in visual neuroscience, specifically from the ventral visual pathway. And the core behavior of object recognition is thought to be implemented by the ventral visual stream. And so in doing so, basically, it's thought that the VVS acts as a hierarchy, a stack of cortical layers, whereby data enters at the retina and travels up the stream. And each stage is a distinct population representation, representing the original input in a more and more abstracted and variant and usable form, whereby irrelevant details are discounted along the way. And so by the time information has flowed to the top of the ventral stream in inferior temporal cortex, or IT, it's been transformed into an effective basis for object recognition judgments. And similarly, convolutional neural networks work by taking an input image and applying a sequence of spatially repeated linear operations interspersed with local nonlinearities. So each ln chunk can be thought of as somewhat modeling what a single brain region in VBS might do. And these can be viewed, one, as functional models, in that they can perform the object recognition behavior along with some other visual recognition tasks, as well as structural models, in that each layer has a set of artificial neuron activations that can be mapped to biological neurons. And sort of the canonical result here, when by Dan and Hung in 2014, was that when we performed this mapping, we see that these networks perform the best, in fact, strongly surpassing prior sort of hand-designed feature models, with a strong correlation between performing the tasks and IT neural predictivity. But it's not just the top of the ventral stream that we can predict well. We can even get fits to intermediate areas like V4 solely by optimizing for the task readout at the top of the network. And moreover, the hierarchy is also obeyed in these networks, in that intermediate areas are best fit by intermediate layers of the network, and higher areas are fit by layered layers. And beyond just predictivity, as a byproduct, we can even use these encoding models to actually control intermediate areas, for example, like V4, far better than hand-designed optimal stimuli. And these models don't just apply to the visual system, they can also strongly predict other sensory areas, including auditory cortex, whereby deep neural networks are trained to categorize words and genres better, predict primary and non-primary auditory cortex and alternatives. And this type of approach is not just restricted to CNNs and sensory areas, but also to RNNs and motor areas, as in David's work, where RNNs can be trained to mimic muscle activities as a function of condition. Now, looking back on the sensory system results, we can kind of break down this modeling strategy at a higher level into a few components. So first, we have to formulate a comprehensive model class, like CNNs, and then optimize them on a ethologically relevant tasks, such as categorization. And in the motor system, this was basically done with RNNs as our model class with the task of motion generation. So common to both is a somewhat more general principle, it seems, of goal-driven optimization. So the three general kind of primary components that might make up a goal-driven optimization would be the architecture class of the network, corresponding to our hypothesis for the circuit, the loss function and data set that jointly are used to train the network, corresponding to our hypothesis for the behavior that, say, the brain area under study subserves, and finally, the learning rule, which is a means by which to obtain suitable representations given an architecture and class and task. So in the rest of this presentation, I'll demonstrate some applications of this framework across multiple brain areas and species to study the roles of recurrent processing in the primate ventral visual pathway, mouse visual processing, as well as applying this to some non-sensory problems such as heterogeneity in rodent medial and entorhinal cortex and facilitating biological learning. So to start with, I'll start by discussing some of the first work I did in my PhD. And what I'll talk about in this section mainly will have to do with modulating the architecture class. And it comes from two publications, first in Neuroscience, co-led with Dan Baer and Yamins, and some recent work that we published. So the motivation for studying this coming back to kind of this observation is that while CNNs are actually inspired by visual neuroscience, they lack an important feature. Namely, they lack feedback connections. And as a result, when we map these CNNs to V4 and IT responses, or in general, they can only really explain the temporal average of responses post-stimulus presentation. But the data itself is actually highly reliable at a much finer grain of 10 millisecond bins. And we can see that, for example, recurrence itself in a model can actually give rise to dynamics. And there's actually a wide range of possibilities for what dynamics recurrence can give rise to. So for example, here, in a purely feed-forward network, purely feed-forward networks will exhibit very simple dynamics where basically when the image is on, the response is on, and when the image is off, it's off otherwise. And we can start to get more interesting dynamics by introducing bypass connections where the model will turn on earlier, more interesting with local recurrence at each layer, as well as a combination of long-range feedbacks as well between layers. So the kind of larger question is, if the dynamics we observe result from recurrence, how can we assess what they're useful for? They're not just in visual cortex, but they're really a more ubiquitous motif. But for us, we're just going to stay focused on the context of core object recognition. And so what we'll do is we'll broaden the modeling class of CNNs to conv-RNNs, or convolutional recurrent neural networks, which extends the calculation temporally and has both local and long-range dynamics optimized for an object recognition task. And so we want to basically be able to do this at scale in order to explain variance in neurons, since given that we already know ImageNet enables an explanation for time average primate VVS responses. And we use conv-RNNs to address this with the right timing, where each time step is basically treated as a propagation between layers, so 10 milliseconds. So where should we begin when it comes to kind of building these more recurrent models? Well, we first want to start with maybe a reasonable feed-forward base model into which we'll basically, the idea being that we'll implant recurrence into it. And based on the timing of the forward stimulus pass, we think somewhere in the range of 10 to 12 feed-forward layers is reasonable, given that it takes about 10 milliseconds for the stimulus to go initially through each cortical area after post-LGN. And if we make feed-forward models of this depth, where each layer of the model represents one brain region of VVS, we replicate this result that brain areas that are sequential in the visual hierarchy map onto sequential layers of the model. But the crucial point is of course, if the models are much shallower or much deeper, the best fitting layers for a given population of real neurons will get squished in or stretched out respectively. So we think that basically models of this depth capture the structure of the feed-forward component of visual processing. But there's another issue, which I think comes from really the standpoint of behavior when using CNNs of this depth to model VBS, which is that basically these networks on the left are far from, say, state-of-the-art performance on computer vision and probably from human performance as well. And so given that there's a big divide, say, between this model and a model that's, several times deeper than it, the natural question is, can we fill this behavioral gap or this performance gap with recurrence? And is this what the ventral visual stream also is doing with its use of recurrent connections, at least during core object recognition behaviors? And we can start to do this. So we can add kind of very simple decays, temporal decays at each layer, and optimize it for the task. And that doesn't really help. Now we can kind of resort to the ML literature and basically construct and implant convolutional versions of various recurrent neural networks that we can try. And kind of surveying that literature, we start to see a few motifs emerge. One of direct pass-through, where at the first time step, a zero initialized hidden state basically allows a feed-forward input to pass on as like a single, acting kind of like a single LN block, as in a standard feed-forward CNN layer. And then the other principle, which is somewhat complementary to it, of gating, in which the value of a hidden state determines how much of the bottom-up input is just passed through, retained, or discarded at the next time step. So, for example, your standard kind of feed-forward resonant block has a direct pass-through but no gating. Likewise with the simple RNN, but an LSTM has gating but not direct pass-through. And so we can implant those into the networks and get some improvement, though we really do rack up a large number of parameters here just by adding these recurrent cells. So there's certainly a lot of improvement from just that. So we reasoned that basically we would try to combine these principles. And so we had seen some evidence in shallower networks that developing novel kind of convolutional recurrent cells with both principles was most performant as a function of unrolling the network in time, not just by adding parameters at the first time step. And so we ran an evolutionary search with it. So what we did was we parameterized the nonlinear operations and convolutions within each cell block. And each blue dot is a model. So there's about a few thousand models there. And we tried to search over both the discrete parameters as well as the continuous hyperparameters of learning. And thanks to David for securing the resources for us to be able to do this. And what we find is kind of at the end of the search, we observed overall across sort of the best performing candidates, a strong selective pressure for both gating and bypassing, as well as parameter efficient depth separable convolutions. Which it's just a technical term for saying that there's no mixing between spatial and feature parameters. And adding these certainly helped. Minimal is this sort of temporally not unrolled control, and then we unroll it further. And then we also found additional gains between certain choices of feedback connections, and also other choices hurt more. So by implanting the ones that helped, we can match the performance of much deeper models with both global and local recurrence, but having far fewer parameters and units all along the way. So what we did was then, okay, we built a model. So now we want to actually see if we improve any explanation, at least in this domain. So one metric that we compared to beyond neural dynamics was behavior. And these are primate object solution times, which is basically the time in which both the IT and the model solve an image. And crucially, both conv-RNNs and CNNs can be compared on this metric. And the reason why that's important is it's easy to say you've built a better model by having a metric where the thing you're improving is scored 0 on it. And people do this. Yeah. It sounds, yeah, but really we wanted to actually say, CNNs do have dynamics. Every layer has some propagation. So we can actually assign timings to those into which layers solve which images first. And so what we do is we basically just look at the first time in which the model and IT neurons surpass the primate D prime on each image on the set of commonly solved images for both. And so very deep CNNs will have more temporal expressivity by virtue of having more layers. And so one comparison was to be able to compare very deep CNNs with actually shallower CNNs with recurrence. And just kind of in a nutshell, we found that CNN conv RNNs of intermediate depth, so this 10 to 12 layers that we started with, better match OSTs even more so, definitely more so than their feed-forward counterpart of the same depth, but even more, but also more so than maximally expressive feed-forward networks of hundreds of layers. And these conv RNNs also best match neural responses. So the ones that actually best match the OST the object solution times on the x-axis also have the best average neural predictivity on the y-axis on both early and late time points. And to kind of get a better grasp on like what are the features of these networks that allow them to best match these object solution times is that one thing is we plot basically task performance versus network size, either as measured by parameter count, so number of synapses, or by unit count, so number of neurons that are used. And what we found is that actually size is measured by number of parameters isn't a super constraining thing. There's actually a lot of choices of parameters to obtain the same task performance. But deep feed-forward models have many more units than conv RNNs do for a similar task performance. So what we see is that the conv RNNs actually, on the bottom, best match IT's OSTs by maintaining a sort of trade-off between both unit count and categorization performance. So in summary, at least takeaways from this part are that recurrent motifs that have both direct pass-through and gating lead to matching task performance of much deeper CNNs with fewer units and parameters. Unlike very deep feed-forward CNNs, we can get more anatomically consistent matches with these intermediate depth conv RNNs. And the conv RNNs that have high task performance but small network size as measured by number of neurons are most consistent with the temporal evolution of primate IT object identity solutions. So what this suggests, taken together, is that recurrence in the ventral stream, there seems to be basically consistent evidence that it's extending feed-forward computations by mediating a trade-off between task performance and physical size being able to fit during core object recognition. So what I'll talk about next will mostly have to do with modulating the architecture and behavioral goal together. So thus far I've spoken and motivated a lot of this about primate vision, but given that rodents are commonly used as a model organism in experimental neuroscience, we also wanted to be able to apply goal-driven modeling to this system as well, starting with the visual system, and then in the later part, we'll apply it to modeling rodent medial entorhinal cortex afterwards. And this is work that was co-led with Nathan Kong and is out as a preprint right now. So we'll start with the architecture and ask what's a good one. Prior results have used actually very, very deep networks that are actually primarily suited to primate vision to try to match responses in mouse visual cortical areas. And at a first glance, it seems to suggest that mouse vision is representationally deep because it's best predicted by intermediate layers in this network. But if you actually go back to the anatomy, if you just look at the anatomy itself, this is from the Allen Mouse Brain Atlas. Viz-P is the standard for V1, and then all the intermediate and higher visual areas are not actually hierarchically stacked. They're kind of like branch off of V1, and it's far more shallower network. So there seems to be actually very little hierarchy in contrast to the primate ventral stream. And even this anatomical observation has functional bearings as well, in that if you just look at the split half reliability of neurons, there basically seems to suggest like roughly 3 to 4 stages of processing just based on the timing alone. So, and moreover, like there's just low predictivity between model layers and mouse visual areas. So you might think, oh, well, goal-driven modeling doesn't apply here or something. A natural question I think for us was, well, could we try to improve this correspondence with by fixing a few components here and really adapting it more to the mouse? So we'll start with the architecture. And what we found was that as we score a bunch of different architectures on each of the areas that are standardly used CNNs, we found that Alexand was among the best CNNs typically used for modeling primate vision, 8 layers as opposed to 18, 16, or even 20 in some of these mouse nets that people have invented. And AlexNet is still around 8 layers, pretty hierarchical, right? It's not a shallow network. So we actually just wanted to see if we could build even shallower models closer to the 3 to 4 stage processing that we saw earlier. And if we look at model predictivity across visual area, what we see is like roughly the first 3 to 4 layers are best predicted of AlexNet for each area. And so what we did was like we took AlexNet as a base model and grafted the first 4 layers into networks that also have a combination of parallel streams and dense skip connectivity, which is a very well-established feature in the mouse connectome. And what we see is that we can get similar or comparable performance with half the depth of an AlexNet in terms of fitting the data. So the next question was, now that we've modulated the architecture class, can we modulate the loss function? And so typically, right, what we've been doing with, at least in the primate case, has been supervising networks with 1,000 category labels. You can already make the argument that that's unnatural for primates, but it's super unnatural for mice. Both the type and the number of categories is unrealistic for mice. So what we considered was actually more unsupervised objectives. One of the most notable, I mean, there's a lot of choices here, and I can talk about them, but like autoencoding, for example, but that tends to give very weak representations. And some of the most successful versions of this came out recently where basically it's a very simple idea. You just try to maximize similarity on 2 different views of the same image and minimize similarity between different images in your embedding space. And what we see is that basically in green is the unsupervised version of one of these models. We see that using an unsupervised contrastive objective further improves neural predictivity. But there's kind of one last component of behavior, which we've ignored thus far, or I haven't really just talked about, is the data stream, the inputs, the actual input device that goes into the model. Prior work looking at basically say 2-way alternative force choice tasks comparing ratings of visual frequencies finds that evidence for like an upper bound on mouse visual acuity to be about half a cycle per degree. And then on the right is primate visual acuity over development, which is generally much higher. So one way we encoded this, and this is definitely not the most sophisticated way of doing it, but we just wanted to see if downsampling the images did anything for us. And what we did was actually we sweeped over training. We trained different instances of the same network, but on different image resolutions on these unsupervised objectives. Both these kind of multi-stream models and Alex and what we find is that effectively reducing image resolution during training improves neural predictivity across all visual areas, with the peak occurring roughly around 64 by 64 pixels, which is within the physiological parameters. And so if we put this all together, a shallower architecture, an unsupervised loss, a contrastive loss, and lower resolution inputs, what we see is that, so the previous model, this distance from the previous model, which I showed at the start of this presentation, which is a VGG16, we can basically get a much more significant improvement, close to how similar the animals are to each other, with a shallow, low resolution unsupervised model, as compared to, in contrast to a deep category supervised primate model. And in fact, not all unsupervised, this is sort of kind of diving into it a little bit more, not all unsupervised algorithms actually improve neural predictivity across visual areas. You could try, we tried other unsupervised objectives like autoencoding, rotation prediction, and even like a stand-in for mouse, like whisking, like depth and normals estimation, which is more supervised but more reasonable. And really the contrastive objectives help the most. And in fact, doing too well on ImageNet, the contrastive ones are the best among the unsupervised versions, so they have to be strong representational learners. But doing too well on ImageNet does not improve neural predictivity, which is kind of consistent with this intuition we had about it being, the supervision being not a good signal, but the data set might be. And moreover, increasing the number of parameters tends to be associated with worse neural predictivity. So the takeaway is that mouse visual cortex, at least insofar as the data we've had to compare to, across visual areas seems to be best captured by three ingredients of a shallow architecture, a contrastive unsupervised loss, and a lower resolution input. And maybe the interpretation that's tied to that quantitative assessment is that mouse visual cortex might be a more general purpose machine utilizing its limited resources to perform a variety of visual tasks. So learn more general purpose representations. And this is really in stark contrast to, in the primate case, a much more deeper, high resolution and object category specific visual system. And an interesting kind of maybe point jumping off from this is that the generic nature of this behavior could potentially be used by other sensory systems. So now thus far I've spoken about both primate and rodent vision. And so we wanted to also apply this to a non-sensory area, to apply goal-driven modeling to a non-sensory area. So what I'll talk about in this section will also have to do with modulating the architecture class and the behavior. And this is work that appeared in NeurIPS this year in collaboration with Lisa's group. So kind of going back to a motivating animal behavior that we want models to be able to do is that, the ability to basically self-localize in the environment and to update one's position on the basis of self-motion are core components of navigation. And animals have a very remarkable ability to do that. And central to this ability is a spatial representational system that'll contain place cells in the hippocampus, which become active when an animal enters a particular place in its environment, as well as cells in medial entorhinal cortex, the most famous being grid cells, which exhibit periodic firing fields that form a regular triangular grid across the environment. And these are all thought to be generated by path integration to serve as part of a neural map of self-location. But grid cells aren't the only cells in medial entorhinal cortex, and this is in data that Caitlin Mallory collected. I just quantified percentages. They make up about 20% of the MEC population, and in fact, the more canonical grid cells that we think of are a far smaller percentage than that. This is with a very somewhat liberal notion of what a grid cell should be. Order cells, which fire in the presence of an environmental boundary, make up about 10%. And then you have a bunch of these other blobby cells, which have kind of unconventional and more heterogeneous tuning to navigational variables and are less obviously well described in terms of simple words in terms of their spatial tuning patterns. So we wanted to ask two questions. How might we characterize the response patterns of these cells? And really all the cells in the circuit, not just heterogeneous cells, but to include them as well. And what functional role do these cells serve in the circuit? So one modeling insight that came from prior work was that if you trained an RNN to path integrate, the internal kind of representations that are developed as a function of end-to-end optimization start to at least qualitatively resemble grid cells and other cells in medial entorhinal cortex. And it's just interesting that you could kind of get this suite of cells through end-to-end optimization rather than hand design features. And so I guess one motivation for us was, can we use these models that to provide an understanding of the circuit, and meaning that we'll tune these two knobs that we have available to us in the model and see a behavior in circuit and see if these structural and functional constraints, such that when you optimize for them, give rise to response profiles that actually quantitatively match MEC. And the reason why we want to actually quantitatively compare these models is that it's not obvious that these two heterogeneities ought to be the same by eye. And in fact, you could have many more ways of having heterogeneities, much more so than, say, grid-like responses. And in fact, what we'll show is that it's the case that not all models are equal. So how might we go about actually defining similarity between sets of response profiles that we can actually describe in words? So what our model is overall is that a model should be as similar to the system as it is unto itself. And what we mean by that sounds kind of Confucius-y, but what it really means is just that, suppose we have recordings from populations of several animals, and let's say an artificial animal on the right, the model, both the model and the animals produce a set of neural response profiles over a set of stimulus conditions. Now, this is a little abstract, but in our case, we'll, for example, consider conditions over spatial bins and consider the rate map representation of each neuron's response. And in theory, this can also be other trajectory variables like speed and head direction. But to kind of make the comparison of vision, these stimuli would correspond to actual images. And then we can draw a set of arrows. And what they mean is that basically when we compare models to animals, we basically use the same transform between units in different animals as we do when we compare the models to each of the animals. So we basically will treat the model as an in silico stand-in animal, but force it to use the same transform that makes different animals similar. So then the question is what should this mapping be? And kind of just to abstract, to step away a little bit, what we see is that ultimately we use the variety of linear transforms, and we find that a form of cross-validated linear regression that's constrained gives the best similarity between animals. But crucially, between all transforms that we use, we actually found that more heterogeneous cells, which are blue, are reliably have a reliable consistency across animals. So that suggests that they're actually reliable modeling targets to begin with. It could have been the case that they're really unreliable, in which case, I don't know if we would expect the model to, if we should have the expectation the model should be good explainers of them. So going back to the models now that we've established a target, we are first going to start with behavior and consider a spectrum of tasks. So we consider a few variants of the path integration task. And we can basically think of them in this somewhat more generalized framework, which consists of a network getting velocity inputs, followed by a type of intermediate MEC-like layer, and then maybe a place cell-like layer from which one can eventually decode ground truth Cartesian positions in X and Y. And so the simplest model actually here would be the inputs, namely the velocities that we get in. And so if our quantitative methods are accurate and the neural data allows us to assess this, we should be able to this should be basically a bad model. And so like in vision, the reason for this intuition is in vision, this model would be akin to asking how well do the pixels explain responses in visual cortex? And the answer is not that well. Now the next class of models consists of taking neural networks more seriously, whereby the MEC network gets velocity inputs followed by an intermediate representation, which is then followed by supervision by these idealized place cell centers from which we can decode ground truth positions. Another kind of task is to basically be able to directly supervise on positions and forget about this intermediate place cell representation. And we'll demonstrate that this is a quantitatively worse idea. And then finally, one additional model, it's not really a physical model, but it's more of just an output-based model, which starts with these place cells and performs a low-dimensional factorization on them. And it's really the main reason to compare this is it's a theoretical model that's motivated by mainly trying to obtain representations that are biased towards grid cells specifically. So we wanted to also be able to compare this. So now we'll turn to the other knob that we'll tune, which is the architecture class. So what are the possible circuit constraints that these neural network models might have? And we consider a variety of RNNs, so very simple path integrators to those with a single level of learnable modulation as to what information to store over time and what to discard. So for example, you can imagine this might be useful for path integration over longer timescales. And then we can increase the amount of gating units, as well as modulate the output nonlinearity of each model. And we have a variety of choices from those which saturate within a fixed limit to those that are purely non-negative to completely linear. And so if we start with I'll just break down this plot. If we start on the right is the internet, how well animals explain each other. What we see is that kind of as the main result is that the best kind of task optimized models can actually approach this intra-animal consistency. And this is with a single gated RNN, this is the UGRNN with a ReLU nonlinearity. And we can actually further dive into the ingredients that make it a good match. One thing we see across all models is that having a non-negativity constraint plus some amount of gating aids in generalization across environments. So we train networks both in a larger environment and in the same environment, and the ones that are trained in a different environment generalize well when they have a non-negativity constraint. But it's important that a non-negativity constraint doesn't saturate either. Something like a sigmoid isn't as desirable, even though it's non-negative. And the models themselves, more crucially, add a lot of predictive power to their velocity inputs. And having the output place cell supervision provides better generalization over direct supervision of Cartesian position. So having this kind of intermediate representation seems to be desirable, at least in matching the data. And that intermediate representation that we're using is also not a good predictor of MEC, which is somewhat of a sanity check too. And furthermore, dimensionality reduction on place cells is not a good predictor of MEC either. So this is sort of more theoretically motivated model. And beyond linear regression, we can actually compare to unit level metrics. For example, the distribution of grid and border scores. So in this case, the gray is the data, the grid score distribution. And what we see is that the best model class in terms of neural predictivity also matches grid score distribution in its own synthetic population. And the low rank model is actually too biased, as we kind of understandably, towards grid-like units, making it a worse match. And this sort of best RNN architecture without the intermediate place cell representation to supervise over is actually biased in the other direction towards too many non-grid-like units. And this best model best matches the grid score distribution across other. And we can also do the same thing with border score distribution and see the same result.
Can I ask a quick question?
Oh yeah, sure.
So in that model where you optimized, I probably don't understand something, but where you've optimized you use the neural data, something about the grid structure to optimize the model.
No.
You did not do that?
No.
Okay.
It's just after you task optimize the model and just compute this metric and compare it to the distribution. It's not optimized for the data at all.
I'll follow up then. Thank you.
The kind of last result in this section, we compared to 1D VR with Qs and saw a similar result. But we wanted to consider some other scenarios beyond open field, and one was this very well-known result from Will and Kia in 2019, whereby having a reward in your environment leads to remapping in MEC. The reason for that we want to capture this is, well, the same circuit is producing a change when a reward is present, and if we want a correct model of the system, we ought to try to replicate it in as many scenarios as we can. And one inspiration, though, from animal behavior that we saw was that actually animals tend to take a more rapid, direct path to the reward zone when a reward was present in this environment than in the open field condition on the left. And what we found was, kind of in summary, was that actually a reward biased path integrator that modulates its trajectories to go directly to the reward zone, like the animal, best captures MEC responses across both conditions, close to the inter-animal consistency, even though it's not trained in the same environment. And on the right, it captures the remapping at the unit level. So the data in gray, this is just a correlation between the rate maps of the same neurons and doing the same thing in the model across conditions. And moreover, it's actually an further observation that actually a lot of this reward remapping is strongly input-driven, but not completely. So if we took the exact same network and didn't bias it towards rewards during training, but just evaluated into two different conditions, we actually see a lot of that predictivity. And this is something that's kind of nice to be able to check in the model that's hard to check actually in the data and provide some further interpretation. So the takeaways of this section are just that heterogeneous cells are reliable targets of explanation between animals. This sort of classic theoretical model doesn't quantifiably explain all the data, even if you increase the number of factors as far as you can. There's differentiation at the level of task, in that being able to train on this path integration task gives you a higher correlation than with NMF loss, but further having an intermediate place cell representation is important. Having a specific type of circuit architecture and nonlinearity as UGR and ReLU gives the best match overall and approaches the inter-animal consistency. And I think the broadest takeaway is that effectively, maybe a way of sort of probing MEC is, rather than focusing on a few canonical cell types, it actually forms more of a continuum, in that formed by a process of biological performance optimization. And the cell types themselves are actually unified in the same path integrator network. And then the last, I'll just finish up with this last section, which is biological learning. And I haven't touched this at all in everything before this section. So what I'll talk about in this section will mainly have to do with modulating the learning rule component of goal-driven modeling. And this is work that's spread across 3 works. And the first paper in this section is work that was co-led with Dan Kunin and Javier Sagastoy-Brena. And so the main idea is, we've been using backpropagation so far in all these networks, but we can actually think of a spectrum of credit assignment strategies when looking at the plasticity rules proposed in the literature so far. Simple Hebbian learning, which dictates that a synaptic connection should strengthen if a presynaptic neuron reliably contributes to the postsynaptic neuron's firing. But it's very hard to make meaningful changes to its input because it doesn't consider downstream effects. You can also consider other things like perturbation-based methods like node or weight perturbation, which basically use a measured change as a global scalar reinforcement signal to accept or reject a perturbation. And then finally, backprop, which computes synaptic updates to most quickly reduce the error. And this is more of like a vector error signal as opposed to scalar one, like in the prior cases. And so we can think of backprop and perturbation algorithms falling along a spectrum with respect to the specificity of the synaptic change they prescribe. And algorithms, as a result, will learn at different speeds. The less specific you are, the slower you tend to be. But despite the efficiency and usefulness of backprop to get us good representations, a major issue in its biological plausibility, without even comparing to data, is that it just requires the feedback network here to perfectly know the weights of the forward network at every step in learning. And this sort of perfect symmetry in a real biological circuit, at least, just seems hard to justify on the face of it. And we call this the weight symmetry problem. And so what we did was we considered relaxing this weight symmetry by replacing the weights in the backward pass involved in generating the gradient to form a pseudo gradient with another matrix B that's randomly initialized. And the most kind of famous or well-known choice of this is feedback alignment, whereby B is just held random to a fixed matrix. And a few years ago, it was shown that that these feedback alignment can basically do well on as comparably to backprop on simple tasks like CFAR. But the problem is that as you go to ImageNet on the right with the black bar, the gap widens between a backprop trained network and something trained with these alternatives. And the reason we care about this is we know that we need difficult tasks like ImageNet to be able to explain neurons, explain variance in neurons. So if our learning rule can't yield these effective representations, it's very difficult to then consider it and its application to the architecture and task as a viable candidate to be compared to. So how might we go about ameliorating this issue a bit? Well, rather than asserting that the backward weights are completely fixed and random during training, we can allow them to change over time. And so one recent proposal was that this weight mirroring procedure whereby in the backward pass, you can update B so it eventually learns to align with the forward weights. And what we found in our hands was at least if you take different variants of ResNet, different architecture CNNs, and train them with the same metaparameters of learning with backprop, you get very consistent performance. But if you do the same thing with this weight mirroring procedure, the parameters actually fail to transfer. And you can consider this a little, difficult to stomach, I guess, because from at least a semi-evolutionary perspective, it would be highly disadvantageous for a learning algorithm to have to be retuned from every scenario to another scenario. And this is not an issue that we see in backprop at all, despite getting competitive performance for one model architecture class. So what we did was we implanted various other options of learning circuits to form as a form of regularization to search over. And we found different alternatives that scaled differentially. And we found specifically that having specific normalization operations during the process of learning was beneficial. And so from a theoretical standpoint, this is a useful existence proof that we can get a learning rule to at least scale across architectures with the same parameters. But from a scientific standpoint, a natural question is, are any of these learning algorithms more consistent with current neural data, such as the data, for example, we might typically compare CNNs to? And what we see is that actually, if we take, say, ResNet 18 and then vary the learning rule as opposed to varying the other components, so just fix the task, fix the architecture, but vary the learning rule, when we map to temporal averages in this adult macaque, we can't really with this data separate anything other than feedback alignment and everything else that scales well with backprop. And this kind of highlights both the importance of a learning rule scaling on difficult tasks, actually be able to perform the behavior, but also suggests the need for more temporally precise measurements, likely when an animal is, say, learning. But what should that experiment even look like? What should those measurements be? And we can measure synaptic strengths reliably in vivo. This is very difficult to do, as Josh Melander has done and done with hundreds of synapses in vivo. And we can obtain descriptive statistics about these synaptic updates and see differences, say, between inhibitory and excitatory cell types. But can we go beyond that and actually beyond, say, these descriptive statistics and actually be able to separate different candidates? And so, given that we didn't have any experimental data, we decided to just kind of generate our own. So we basically took a virtual experimental approach to this question, with the goal of answering whether it's even possible to generically identify which learning rule is operative in an artificial neural network across a range of learning rule types, architectures, and loss targets, and what neural observables in those networks are most useful in making these identifications. And really, with an artificial neural network, you can measure anything you want to know, and the ground truth learning rule is known that we train the model with. So if we can't do it in this setting, it just seems very unlikely to be able to do it in a real experiment where we have limited and less precise measurements. And what we'll show support for is that measuring postsynaptic activities, you can measure synaptic strengths and get insights, but measuring postsynaptic activities on the order of several hundred units may provide a good basis on which to identify learning rules. So our approach was basically generate high variation virtual experimental data across a wide range of scenarios. So we have about 1,000 networks that were trained. And then we basically just turn this into a very simple classification problem and test them on held-out data and consider various types of generalization that simulate different forms of experimental realism. And what we do is, as a network is training, we basically concatenate trajectories from the weights, activities, and layer-wise changes and analyze what features are most meaningful. And at least from the get-go, when we have perfect access to everything, at least we can separate these learning rules effectively with a suitable sort of old school kind of like classifier. And now what we wanted to do is basically knock things out and see what would happen. So the reason for this is just to keep in mind that you might imagine that different experimental tools have different limitations, either based on number of units you can record or precise timing as well. So to account for these kinds of trade-offs, we basically modeled measurement noise as an additive white Gaussian process added to model units. And on the top right was the perfect information setting that we had before. And what we see is that actually, if there is a reasonable amount of noise and subsampling on the order of several hundred to several thousand synapses, a noisy imaging strategy of, say, synaptic strengths could be rendered ineffective. But recording from the activities on the order of what might be achievable with electrophysiological techniques could form a good set of neural data to separate learning rules. And of course, recording more helps, but it doesn't seem at least necessary to record a majority of units to be able to do this effectively. So at least in this learning rule component, I'll just kind of end here, is that we don't necessarily need to enforce weight symmetry strictly for effective learning, and we can actually build these performant relaxations with the appropriate layer-wise learning circuit. And to be able to then further differentiate learning circuits as candidates of learning in a real brain, we can actually identify them only on the basis of aggregate statistics of various measures of weights, activities, or layer-wise changes. But overall, we find that the network activations are the most robust to various amounts of experimental realism, and others that I didn't actually show in this just for the sake of brevity. I'll just end with some broad takeaway conclusions. So from the first part of modeling recurrent connections in the primate ventral stream, to basically, we see evidence that they're there to enable a high-performing network to fit in Cortex that attains computational power through temporal rather than spatial complexity. And when it comes to modeling rodent visual cortex, we found that a low-resolution, shallow network that makes the best use of the mouse's limited resources to be general purpose seems to be the best quantitative match to that system. In MEC, we found that heterogeneous cells aren't functionally segregated in any way from classical cell types, but form a continuum of cells within a single unified network architecture that can encompass both through end-to-end optimization. And when it came to building and identifying learning rules, we found that weight symmetry isn't required for effective learning, and that we can differentiate these hypotheses of learning circuits from imperfect recordings of postsynaptic activities. And with that, I'm grateful to my wonderful collaborators across these projects, without whom none of this would be possible. And then also my funding resources from both the Neuroscience PhD program and MBCT. And I'm happy to take a few questions now, and then I'll go into extended